to Harry, Sydney, and the Cell Next Door. This is episode 3 and our last episode, Can We Teach Enzymes New Tricks? So, coming through from last episode, think about this. Your favorite shirt, your leggings, or even your water bottle, they feel solid, but they're made of long chains called molecules called plastic. And plastic, they don't just disappear. They're designed to last.
Right. So, you know how they know how to break down wood, food, or complex oils, but polyester, nylon, PET bottles, these things are brand new inventions in Earth's history. So nature's never ever had like a reason to evolve things or like enzymes that can eat them.
Enzymes, by the way, are proteins that make reactions happen faster. They're the reason that your bread rises, the reason that our stomachs digest lunch. But here's the catch. They're very specific. An enzyme that digests starch won't touch protein, and one that works in your stomach won't work in your laundry detergent.
So basically, they're like a very picky eater. You could think of it that way. And when it comes to plastic, most enzymes don't even recognize them. It's like trying to open a lock with the wrong key.
Scientists did, however, find some enzymes that can attack plastics, like PETase. It can cut PET, the plastic that is used in bottles or clothing fibers, but PETase is weak, so it works slowly, and it also only functions at certain temperatures, and it also falls apart under industrial conditions.
So PETase is really more like a prototype. It showed us that enzymes can break plastic, but not well enough for real-world recycling.
So that's where protein engineering steps in. The idea is simple. Change the enzyme's amino acid sequence, which is the recipe that builds it, and see if we can make it stronger, faster, and tougher.
So there's two main strategies. The first would be irrational design, and that's when scientists look at the enzyme's 3D structure and they predict which part controls things like stability or activity, and they make the targeted changes. It's like fixing a machine with a blueprint in front of you.
The second is directed evolution. That's more like nature's trial and error, which is sped up in the lab. And you can make slightly different versions, thousands of slightly different versions of the same enzyme, test all of them, and then keep the ones that perform better. And then over these several rounds, you end up with an enzyme that's way more powerful than the original.
And this isn't just a theory, right? Using these strategies, researchers have already engineered enzymes that can break down PET hundreds of times faster than PETase alone. And at higher temperatures, that means they're closer to working in recycling plants, not just petri dishes.
And of course, there's a big question: control. You don't want an enzyme that breaks down plastic to accidentally attack natural fibers like cotton, or worse, even interfere with biological tissues like humans.
Yeah. So that's why designs, they always come with like a safety in mind. So enzymes can be tuned to only work under very specific conditions, like a certain pH, a temperature, or with specific cofactors present. That way, they don't go wild.
The big picture here is more than just a shirt or a bottle. If we can turn enzymes into reliable tools for plastic recycling, we could reduce textile waste, rethink packaging, and cut down on pollution that takes centuries to disappear on its own.
It's like a shift from plastic piling up in landfills or oceans to a circular system where enzymes help us take old materials apart and rebuild them into new ones.
And it's a perfect example of what synthetic biology can do, taking something nature already gave us and reprogramming it to solve problems that nature never had to deal with.
Plastics may stick around for hundreds of thousands of years on their own, but with the right enzymes, we don't have to accept that story as the ending.
So this had been Harry, Sydney, and the Cell Next Door. Thank you for coming with us on this journey from closets to cells.
Right. See you next time.